South Korea, and he has completed his B.Pharmacy at the Tamil Nadu Dr. MGR Medical University, M.Tech Bioinformatics, and PhD Structural Bioinformatics from Sastra University, India. He started his career as assistant professor at Sastra University, India, and moved to Konkuk University, South Korea, for his postdoctoral research and postdoctoral associate at Max Planck Institute, Germany, and currently working as senior scientist R&D Pharmcadd, South Korea. He has published more than 10 research papers in reputed international journals. As a computational chemist, structural bioinformatics, he has intensive experience in computational enzyme engineering and drug design, reaction mechanism studies using DFT, QM, MM calculations, and he is proficient in molecular dynamics using commercial and academic software, Desmond Gromax, electron transfer pathway studies, the reaction mechanisms, development of novel computational method peptidodynamics to screen peptide mimetic, skilled at mentoring student projects at the level of master's and undergraduate degree, proven track experience in drug design, pharmacophore analysis, QSAR, MD, experienced in basic molecular biology, biochemical, and immunohistochemistry analysis related to drug design. With this short introduction, now I welcome Dakshinamurthy Sivakumar sir to deliver the valuable seminar. Please, sir.
Okay, thank you very much, sir, for your kind note and welcome note. Okay. Okay, these are some of the institutes which I previously mentioned. And let me directly go into the topic, the bioinformatics and artificial intelligence. If you see this, why this bioinformatics and artificial intelligence plays an important role in drug discovery. If you see the, before we go into that, we will just see the basic definition of bioinformatics. There are several definitions available for bioinformatics. One such is like you can use the biology in terms of macromolecules. For us here, it is going to be a protein or a DNA, and applying the information techniques to understand and organize the information associated with these molecules on a large scales. You can put it into any scale, like if you use genomics, genome level data, you call it as a genomics. If you use protein level data, proteomics, like that, go on. There are so many jargons in bioinformatics and there are many disciplines also. It all comes under one roof. Then artificial intelligence, if you say it is one of the growing field and everybody is talking about artificial intelligence, and it's everywhere. You can, starting from your phone, your computer, your day-to-day life, your Alexa, and everything, right? Now you can see artificial intelligence everywhere in and around you. You cannot skip it. Like, so it also plays an important role in drug discovery also. We can, in general, what it is like simulating the human intelligence in machines that are programmed to think like humans and mimic their actions. In short, you can say like we have to make a system or a model or a machine or robot, whatever you say it is. It has to think like humans and it has to act according to the situation and it has to keep on develop its this thing. That's how we call it as an artificial intelligence. It differs from the program where it is adapting itself to the new problems. Okay, traditionally to find a drug, what and all we use, what a pharmacist will do, or the scientist who's doing in the drug research, what they will do. In order to identify a small molecule which can become a drug in the later stages, they first needs to find it out whether it can able to bind to a specific target. For example, they identify a disease, then they look into the several targets and what is the special target and whether that it can able to target that particular protein or the DNA, whatever, involved in the disease. Then they will have to screen large number of molecules. It will be a library of molecules. Either they can get from library like in-house libraries or commercial library like Zinc, or they can get the PubChem like open source library of small molecules. And they have to identify the potential molecule which can become into a drug. And there are several in vitro like biochemical and immunohistochemistry and so many tests we need to do. And also we need to do a lot of in vivo tests, also cell lines and animal studies, and we have to do the preclinical trials and clinical trials to become into a compound. There are so many stages, right? So this is how traditionally the works. But if you see this, the drug discovery pipeline, how it works is like the first two stages we can group into two stages: drug discovery and development. The first two phases: target identification and lead optimization, which is the most time consuming. In this case, like 2 to 6 years, you can it takes to identify the right target and optimize the lead. Then once the lead is optimized, then we need to make into a next stage. That's called drug development. We need to develop the lead. The lead is here is a small molecule, right? The chemical substance mostly, or also a natural substance. And then we need to go for a preclinical development, and then the clinical trials phase 1, 2, 3. Then we have a regulatory approval. That also takes time. But if you see this in your funnel diagram on your right hand top, you can see the first stage is drug discovery, where you have so many molecules, right? Thousands of molecules, sometimes it's even millions of molecules. And then we will reduce into few hundreds, then further reduce into less than 10 numbers, right? So here we will go into the various stages of the phase clinical trial. Then we'll go to the regulatory approval, and we'll finally come in to get into one. So we have to identify this one molecule from these thousands of molecules. That is a challenging one, right? So okay, this is why it is time consuming, and it is very expensive also. Okay, now let's see how this bioinformatics will help in addressing these issues. If you see this, the first step, that is target identification, we can use here various approaches, like some of the machine learning approaches or data mining. Starting from a literature search, we can do a lot of other ways to get the data. What we need, what are the informations? For example, what is the active site? What is the protein's binding cavity? Everything. What is the characteristics of the protein? Everything we need to find it out so that we can develop a complementary drug molecule which can go and bind if it is an inhibitor, right? So we also have to consider its genetic association also, that polymorphism, which is also important, because we are making one drug which is targeting the disease, not the individual, right? For example, the same drug, if a person is affected by a diabetes and you are going to give the same drug to a person X, Y, Z, which is present in India, US, or Germany, or South Korea, wherever he is, the drug is going to be the same if the disease is the same, right? But the problem is we are all individuals, we are different, right? So there are that also we need to address the issue, and also we need to see that how the protein is expressed, where it is expressed by using an expression profile, like which tissues or which cells it's more expressed, whether it is the same protein can express in multiple organs, whether it is expressed in the liver or the heart or in the kidney, where it is expressed or where it is expressed more, where we are going to target. So this needs to be understood. Then the pathway analysis. Most of the side effects, almost all the drug as of now available in the earth is having a side effect. There is no single drug available as of now has zero side effects. Every drug is having some side effects. Why? Because of we are not specific. Our drugs is not specific only to the binding protein. We said here we are identifying the protein, right? Identify the target protein. But whether the drug is going to bind to the specific protein, the answer is no. It's meant to bind to the specific protein, but it also binds to several other proteins, because we have a 20 amino acids, and the 20 amino acids folds into a different proteins, and the proteins more times have a similar folds and similar cavities, right? So it prefers to bind it to one other. We can bind a efficient drug, but specific drugs also, but relative to the available targets. We cannot find it out the very specific zero side effect drugs. There are one of the important ways you can address this is using pathway analysis. There are so many pathway databases are available, so we can study the pathways and where it's all interlinked, so we can address and how if you target one protein, it is not only related to the protein, it's all interlinked. This may other role also. So we need to understand that role, and we should not affect the normal the process also. Some proteins which may plays an important role in the normal process also. For example, even if you take the anti-cancer proteins like BCL2, which is also important for the normal life, right? So it is only when it is overexpressed we need to control it. Like then the overexpressions we also study by using the expression analysis, like a functional analysis, you can say like. To validation. The next is validation. You identify the target these ways. The next is to validate it. How we are going to validate it? For example, let's take a simple example like COVID-19 is a target virus we identified, and there are several proteins starting from the shell, the capsid protein, and to the to inside the cell, like what are all the proteins? There are so many proteins. We need to identify what proteins you are going to target and why it is important. For example, if you're using a spike glycoprotein, it's one of the protein. It's a surface protein, so this is important for the binding of the its binding to its cell surface receptor. Or you can use the some other envelope protein or a membrane protein, something like that. There are also many other proteins also we can use this one. So it all depends on your significance and also your aim, why we wants to target it. Then the next thing is the hit generation. We need to, we will get the hit. The hit is something like a small molecule, usually a chemical substance, which binds to the target, that's the protein here, and it needs to modify the activity. Usually we will get a lot of hits, whatever the technique you will use. Either you will use some ITC kind of analysis, or you are using some bioinformatics techniques like docking or other methods. We will get hundreds and thousands of molecules. Because after all, again, it works on some of the basic principle like shape complementarity and charge complementarity, which can easily get binds to it. So just only based on the binding or how effectively it is present in the cavity, we cannot say that can be a drug. Yes, it can become a drug, but you cannot say that's become a that's the possible drug like that. So there are several stages which needs to address to get this issue. The next thing is lead. How to make the hit into a lead. The lead is a substance, which the name itself says, right? Lead, it's leading. So the promising hit, which has a pharmacological activity. That means therapeutically is useful, but can be useful for further processing like pharmacokinetics and selectivity, etc. Right? So we need to address those issues also. Not just only the binding or the pharmacological activity is not sufficient. Only if it needs to be more than the pharmacological activity. It should need to be therapeutically effective also. It needs to clear the ADME characteristics. Okay, let's see in more deep about this. The stages is hit to lead. We can use the bioinformatics techniques here. Again, we will use two different techniques. One is called structure-based drug design. It's called SBDD. We shortly call it as. Another one is called ligand-based drug design. You can see here. So what is this ligand-based drug design is when we have the ligand or the lead compound or a chemical substance, we know the structure of this one, but we don't know much about the protein. Then we will go with this method. In other case, we will know about the structure-based. We'll go with the structure-based drug design, where we will know about the protein structure, but we don't know much about the protein information. Or the protein information, I mean, ligand information is not available. So in that case, we can use. Okay, so there may be other cases like when there may be a structural information on the protein itself is not available. So in that case, we can do a modeling. We can model the protein using several techniques like a homology modeling, threading, or ab initio. We can do that, and we can get the structure of the protein. And then we can, or also we can go with the experimental techniques like X-ray, NMR, or electron diffraction studies. And also we can go with the probable known actives, we can get there. Now, we will get a hit of compounds, right? So we'll get a pool of compounds. We'll have, again, hundreds and thousands of compounds. Now we'll go with some bioassays, various bioassays you can do. Then we can do synthesis and we'll get the lead. This is the general traditional technique. In the structure-based method, what we'll do is like, first we need to find it out. We have to do the docking-based visual screening to screen whether the drug can able to bind with the several small molecules. The small molecules can be able to bind with the protein. Then we can able to find it out something called pharmacophore. In that pharmacophore, we will see that what are the key factors which is present in the protein, which is important for the ligand bind, like a hydrogen bond acceptor, hydrogen bond donor, or hydrophobic, like that. Then we can do the molecular dynamic study to see how stable the complex is, the protein and ligand complex we have. And we will see that how stable it is in the presence of the solvent or in the environment. Not always we will use only the water as the environment. Sometimes we will go with the lipid. And then we can also we can go with the de novo design also. So here we can not using any database of compounds. We can make the compounds novel, which is what the pharma industry is mostly like, because it has IP, right? So they can patent it. But if you get some compound from the database, it is already existing, and IP issues will come. Then ligand-based drug design is just opposite to this one. Here we will get your ligand-based pharmacophore based on several compounds, small molecules. We will align it and find it out what are the common features, all these molecules, because it has active. It is, sorry, it is active, right? So we will go with a quantitative structure activity relationship and do a 2D similarity search. Or scaffold hopping is another technique where we will replace some scaffold. Say, for example, you see the ligand example which is shown here. Now, for example, if I can just replace, if this is the main scaffold, I can just replace this scaffold and put it into some other, like biphenyl or something like that. So I can put some other group, like this called scaffold hopping. Instead of replacing the functional groups or the R groups, we can also replace the scaffold. That's called scaffold hopping. Then finally we'll go with the admit, and then we will choose this one. So this is a technique used. Now we'll see this. What is lead optimization? As I said before, okay, just we are getting a heat, and heat is sent to the lead. Now the lead need to be optimized to make into a better drug, right, or a druggable compound. So if you see here, another example in the simplified approach, when the active sites, the lead molecule is bound. But if you see here, it's not a tight binding, right? And the second, the figure, if you see the lead, it's not a very tight binding because if you see this one, there are so much gaps. We can modify the lead molecule to make into a better binding one, so that it will preferably bind in this protein and it won't leave before the action is over. Right, so we can see this one. It's called make, test, analyze, and design method. Like first we have to design using molecular modeling and the techniques. Then we have to make using synthesis. Okay, then you can use computational synthesis also. Then we can test it using various several techniques. You can use bioinformatics here also, and then we can use it, analyze it. Then a real-time example I have shown here. This is one of the example of it's published from the MAP kinase. You can see here the first example shows that how the accuracy is improved. Here you can see here in the first case it is 350 nanomolar. The concentration is needed, the activity, but it is further optimized into a 30 nanomolar compound. How? You can see here there is this molecule, this part of the functional group they have removed, and this one is also maybe we don't know, the structure of the protein is not shown here. There may be some clash with this is there or not. It's not compatible here, so they replace this one or add this group. Maybe a phenylalanine group may be there, and then it can be forming a pi-pi interactions like that, right? So that one gives this one. To make it more stable and make a more strong interaction. I hope so. You can see here. So this is how you can optimize. There are several examples are available like this. The next is about ADME. As you are, most of you are pharmaceutical students, you know about what is ADME, right? So pharmacokinetics, what the body does to the drug, and pharmacodynamics, you also know that. So if you see this, when you take the drug, whatever the route of administration is, either oral or whatever, the metabolism site is going to different, but major route of administration is orally. So we'll concentrate on this. So after absorption, the metabolism, like we will see this dissociation, dissolution test we will do. And computationally, how will you do this is like we have to see this, what is the pharmacological effect, like how the drugs into bind into the probable metabolism enzymes and plasma proteins we need to see how well it is get distributed, whether it is can be able to bind with the albumin or globulin that we will need to see. Then we can also see that what route of excretion it's going to take, renal or fecal, that's also. So these are the normal ways. How will you do that? The drug in the, like, I'm not going to deep into it because you might have studied this in your pharmaceutics. Okay, so what are the various tools and resources are available in bioinformatics, but which you can study all these admin properties online, or computationally is there are some softwares which is listed here, like Discovery Studio Talks, and Mal Inspiration, TopCat, AdMet, KitProp, and many. And once you get the molecular structure, as I said before, using the available machine learning methods, we can calculate the physicochemical, topological, and geometrical and its electronic properties and do a QSR studies to see that what are the factors or the properties which is are the physicochemical parameters which is important in determining the activity of the small molecule for its binding that can be corrected. And then also we will predict the log P, log D, log S, and toxicity. Hope what is log P and log P and D and all, right? The partition coefficient we need to see. And then the metabolism enzymes, there are several cytochrome P450 families we need to see. Okay. If you ask me that, is there any success stories, anything you can do it in computer, whether it's really possible, whether it can be, there are several success stories are available. These are some of the common drugs, which some of you might know, like captopril and enlopril, right? You might heard about this one, and lopinavir and islamivir, right? Many where you can see here. So mostly antiviral drugs are available, anti-cancer drugs, diabetes drugs are mostly concentrated. Viral drugs, which is because very fast mutation happens. So those are all some of the examples of the success stories which is available. Okay. Now, where this artificial intelligence? So using this, we can do a lot of bioinformatics studies to make into a effective chemical substances into a drug molecule. So to turn the chemical substance to an effective drug, we should need both desired biological effects and properties, as I said. Potential. There are many cases where the drug is very potential in lab. Even it works in all the animal trials also, like it goes well with the animals and it shows very good test, IC50 values, everything is very good, but still it's fail in clinical trials. Many happens. Only if you see the statistics, it says only 10% of the drug candidates makes to market even after the phase 1 clinical trial. So you can just imagine how much the failure rate, 90% is failure, right? So the more failure rates is here. So that's where the cost is, that's why it is very expensive. It's 42% of the cost spent on the preclinical and animal studies, remaining 58% is mostly on these pre-studies. So to take the drug to the market, it needs 2.6 billion US. You can just count the figure in Indian rupees, how much it is. That much amount of money you need to spend to get one drug into the market. That's why drugs are it's a market. Still, the pharmaceutical industries are making money out of it. Just you can imagine why they are spending so much money. But still, if they can able to reduce the number, the amount they can spend, and also the time they reduce, it will be further benefited and for the company as well as benefit for the people, because the drug can be available to the patients at the lower cost, right? Because this cost will be is borne by the patients only. So AI, artificial intelligence, has the potential to offer over savings for the drug discovery process by the 2028. It's a prediction. This is one of the example I can say, like they have seen that what are the various clinical trial compounds, which is dated back in 2009, and they recently analyzed in the year of 2017. It is published in 18, where we can see here which phases in the different stages of the clinical trial phases in silico drugs have a good approval percentage. You can see here, this shows that over a year, the percentage of approval is increasing for the in silico the drugs which found mainly using in silico approaches. It's not completely in silico. The in silico is majorly supporting at the earlier stages. Okay, now come to another interesting topic: how AI can help in drug discovery. This is most interesting question, right? As I said, AI is everywhere, starting from your phone, day-to-day activities, everywhere AI is there. and everywhere it's going to be more. And compound libraries, which is exponentially increasing day by day. Pharmaceutical companies are doing a lot of researches on this, and then they'll get a lot of compounds, and they'll develop a lot of in-house libraries, and a lot of chemists are also synthesizing a lot of chemical substances, which you can see in a lot of chemistry papers, articles, research article writes, and then they'll publish it, and then again the numbers is increasing. So AI or machine learning or deep learning, which helps to get the hidden data from these compounds. There are a lot of interesting informations which you can get. So what are all interesting information you can get? Say something like if it is a potential compound which needs to be a synthetic feasibility also, it needs to be increased. Instead, say for example, now using any approach, now you identify a compound as a better compound, but if it is not synthetically feasible or it is not cost effective, a very difficult process, time consuming, then it is very challenging for the pharmaceutical industry to make it to produce in the large scale, right? So if you identify it in very earlier stage, we can just rule out that one, which saves the time and cost. And novel compounds for IP issues. We can using the machine learning approaches to get the novel compounds with all the desired properties. What are all the desired properties? Say, for example, you want to design a compound which should get metabolized only in the liver and which should not have a secondary metabolism, or it should get only excreted by the urinal route, or not for the fecal route, or you want to make a compound which will binds only like that, which is a topical drug like that. So whatever the desired properties you want, you can just feed it, and you can make the compound, tailor-made the compound based on the desired properties which is required. And most interestingly, repetitive task. You might have seen the people who are in the analytical lab. They will do the analysis hundreds and thousands of times, right? So the same set of experiment, they will be, you might have seen people doing in the analytic labs, they will do the same set of experiments repeated over and over again, that they can close their eyes and do the experiment, right? So why the need of human there? The machine can do that, right? So that's how this AI plays the role when you want to do a repetitive task, and we can do. And most importantly, FDA also supports the use of AI in drug development. Okay, if you see here, I'm sorry for this because it works only in the full screen. So this one, the thousands of we have your thousands of images and again. The faster route you can, sorry, it's not working, which can able to find it out better. Here, if you see this slide, you can see there are some strains here, right? But there are some hidden strains here, which is not visible using a normal, humans may miss some of the information here while looking into the microscope or using some of the software. So if we can train the models, and it can able to detect based on the several trained data, which can get the more accurate information or a lot of hidden data from this. So you can find out a lot of patterns which we miss by looking into the slide here. I'm sorry for not able to show this because that GIF image which works only in the full screen. Okay. Okay, what are the strengths of AI? Why I have to use AI? As I said, like one is the time and another one is the cost effective with a lot of the less wastage on the incorrect targets. We can avoid those one, and also it is very speed, and which have multiple benefits, which I now need to save, like manpower, money, etc., and timely reach the market. Like for example, pandemic like COVID-19, where we don't have a regular time of we cannot wait for 12 to 14 years, right? We need it in months, we need it even in weeks, right? People are dying every day, so we need this kind of methods like innovative. Of course, we can say it's innovation, like might develop the sophisticated insights to complex diseases which we miss from the literatures, and biased. The humans are not doing. There is a human bias because most of the time, if you see, when the scientist works in the science lab, when they find some experiment, and remember, as I said, 90% it's failed. It will fail, right? So it's somebody else. It's a brainchild, and they invest not only money. Money is invested by company, but the scientist uses his brain and his knowledge to find the drug. Every stages it crosses, it is like it's very crucial for him. So there will be a bias. So he wants to prove that it is good, it is right. He wants to give justification, right? So there is a human bias always there. So we need to say we strongly believe our intuition, strongly believe our experience, but which may sometimes wrong. So the machines don't have anything. They can just use only without any bias. They don't care whether it is found by a Nobel laureate or a undergraduate student. It treats everything same. So molecular representation, one of the in the AI, if you see, in order to get the model done, we need to represent the model, the molecule into a model, right? To terms, how to represent the molecule. Say, for example, here we have small molecules. The small molecule, how you are going to represent as a model? Say, here, look here. In the chemical structure, we will get the physical property. From the chemical structure, we will get the physical properties, like it has with the soluble or like that, or insoluble like that, or biological property, active and inactive. You can relate this one, right? The compound is having this number of molecular weight, more than 500 or less than 500, like that. So what is the relationship between this? So maybe hydrogen bond donor, hydrogen bond acceptor, for example, Lipinski rule, like that. Then in the computational, in the AI, we can say that we can model it using the input data and endpoint or end data. We can just find a model which is relate these two. See here, like as a travestin, we can see the molecular representation by a computational method where we can use the cheminformatics to develop, to get a lot of confirmations of the molecule which can exist. So we can pick one of this presentation, or we can try in the computationally. We won't just look into only one 2D even or 3D also. We won't just only take one confirmation, because maybe it's a bioactive information if you know the data already, or it can be a energetically lower energy conformer. But in reality, it is not so, right? So it can exist in multiple conformation, and one of the conformation which can be slightly higher in its lower energy can also be a better binding. So many ways you can represent it, like in the original space. We have to extract the features by assembling into in one of the space. Like you can represent this to get features here, or you can this way, or in the other way, or in the neural networks, which you can train it by its own to get features get assigned into one space, like this. Okay, this is another slide which will give you a much better insight. See here in the first example of your small doll or a dog, whatever. The input, we can say, you just by seeing this, you can see, say it is a dog, right? But you need to train your model, the AI, artificial intelligence systems. So how we are going to train it? So we can give some rules, like feature-based detections, like some characteristics like eyes, nose, like that, or fur, somewhere like that. So features will be assigned, or it can some of the patterns which can use like regression methods, which can label it when it has this kind of pattern. This is a dog like that. This is, you can get it from based on the several trained data. Right, and this is another example of natural language processing. So, for example, now you will have your text here. The cute brown fox jumped over lazy dog. So now here we will say that count the word frequencies, how many times the words occur. For example, there is occurred in 2 times, and some of the words is not at all occurred, like that, right? So based on these frequencies, so we will know that we will classify it which one it comes into. Then again, next our compound. Again, we will go with the rule-based feature detection. Like, for example, Lipinski rule, you can take one example I'm telling, like hydrogen bond donor, acceptor, aromatic, nitrogen, molecular weight, etc. And you can put it in the features and represent it. Then the deep learning approaches is further modified or better approaches, where we will see that it is self-trained, you can say, like how it can train itself to get into a drug. Label as a dog. No, here we are not defining anything, but it automatically learns from the features. And then again, here is also the words are counted and then make into what it is, features. Again, the neural networks, which helps to aggregate the information which is available from the structure and assemble it and represent it to get into what is the length representation. There may be errors, but we need to improve the accuracy by giving more training, and then it will improve the accuracy, which can be tested by using the test data. So in order to get this information, we will use something called encoder and decoder. So the encoder is something which will encode the information from to ones and zeros, you can say, and then later we will get a more information. Then finally we will decode it back to the compounds or the structures, which can be read by maybe a human or some other programs what we can use, and also we can detect the properties also. Okay, if you see that how this AI is useful in the medicinal chemistry, synthesis also, which plays an important role. If you remember the some slides before, I have shown this design, make, and test analysis in the admit. So the same here we will use here. So approach called retrosynthesis. Maybe you might have heard about retrosynthesis. Retrosynthesis is in general synthesis is what? Synthesis is when you have two reactants and you will two or more reactants and you will get a product. Reactant A plus B, like Na plus Cl gives NaCl, like that. Retrosynthesis is a reverse way where we have a compound and we break the compounds into several small reactants. So this is useful when we will go for the drug repurposing. Right, we will know from the drug, and also we will know that how we can when you get the compound from the library and then the compound is active. Fine. Now the question is how we are going to synthesize the compound, right? So that's where the retrosynthesis works. Now we will make the possible ways it can be synthesized, and also here you can see how effective it is. Which route will be the faster and cheaper. Here, if I go with this route, it will be more cheaper. Then go with this one, right? So like this. So then we will see this. What are the condition recommendations? Like what are the catalysis, reagents, and solvents is needed, and what is the impurities, whether it is 99 or 90, etc. Then again, we have to analyze it. So this is how we are will greatly useful, saving the cost as well as the time, and also a lot of wasting of chemicals. Okay, retrosynthesis also you can group into two, like one is single step method. If you see, this will be more easy for you to understand. First, we will have a multiple possibilities, right? In one case, we know the products and also know the reactants, but so what are the possible reactant to get the possible methods, like that's how you can use. Put one reactant, like for example C or B, and get the product. This is one step. The another one is a T approach, where we will have a multiple reactants. That means first we will add only B and get AB, then add C, then we will get ABC, like that. This goes on. So this depends on the complexity. Sorry, the compound's complexity. The methods will vary. Sometimes you can easily synthesize in a single step, one part synthesis, or you can go with a multi-step synthesis, like that, right? Like that, same. Then condition recommendation also, we have a reactant and product, but what condition we can get more or less impure, right? So that condition, say for you can do it in the vacuum, or you can do it in the atmospheric, or with the presence of nitrogen. Like that, any conditions, choose the conditions which can predict. And also we know, sometimes we know the condition, we know the reactants, but we don't know the product. That also we can do. That's called forward reaction. Can do that. Okay, so I think I quickly rushed to some of the significance of the AA, and a very brief about how this AA can be trained. But there may be a question in your mind. Bioinformatics, okay, because it is long time it's there, and you have shown several drugs. What about AI? Is there any drugs? Yes, there is one drug which is currently in the clinical trial, which is for OCD, which is HT1A receptor agonist, which is also from the A drug, which is a joint venture from a Japan-based company and UK-based company. They used this Exentia as a model, AA model, they are using. Usually, as I said, these stages will take a lot of time, right? Time consuming. For example, here, even using all the sophisticated techniques, you can come into 5 years. But using AI, they managed to get it in just 12 months. Just imagine, just 1 year is where and 5 years is where. 5 times lesser is not, it's a manpower. You can cost, time, everything is saved, right? How much it is going to be cheaper, and also many things. Okay. So also the AI and drug discovery market are also increasing over the period of time. Even in the time of next few years, you can see this market is going to very high. You can see the drug optimizing and depurposing will be very useful. So globally, where this AI is mostly targeting is, you can see, mostly targeting in the oncology and cardiovascular and infectious diseases, neural diseases, where the complexity is very high and the changes are quick. There it will be more commonly used. Some of the traditional targets, not much, like pain receptors or all these things, which is well known, well studied, not much changes happens. We're not considering. I mean, you can use it, but where it will have a great role is where the, like virus and cancer, diabetes, where the changes are happening very quick, right? So there we will have a great useful, and that's chart shows that how it is true. You might have heard about a lot of pharmaceutical companies. If I ask you, can you name some pharmaceutical companies? You will say like Ranbaxy, Merck, and Glasgow, GSK, everything, right?
நகை வச்சிருக்கோம். ரெசிப்ட் இல்லை, நகை மூட்டுக்கலாமா? இல்ல அந்த நகை வச்சதுக்கு.
Hello. Some crosstalk. Okay, so the non-traditional pharmaceutical companies here are there, which is plays an important role, like a small molecule discovery, like Atombase, Exentia. This is the one. This company is the one which is, and Benevolent AI is also a very AI good company. We are our company Pharmcadd is also plays an is also here, and we can see. And there are companies which uses antibodies and peptides and trial optimization, natural products and microbiome, hit-to-lead optimization, animal studies, PKPD, and patient animal, like this. You can see it's all covered. This non-traditional pharmaceutical companies are obviously AI-based companies are all over, and they are in the different stages of the drug discovery pipeline. Okay, everything has some problems. Even AI also has some own pitfalls. It's mainly what are the possible drawbacks is it depends on the availability of the good data. See, it's just a mission, it's just a model, right? So the model is, the quality of the model depends on what kind of data quality you provide or you feed, right? If you get a good quality data, it's good, because most of the times we will get it from the third parties, like from the published literature or from other labs or some other institutes. If they give the wrong data has wrong mistakes, then the results will also be wrong. And novel molecules which have a limited data available to the researchers, also there is also a challenging. Okay, so thank you for your attention. If you have any questions, please.
Thank you, sir. Thank you for your valuable presentation, sir.
Thank you.
Now the forum is open for questions. Good morning, sir.
Yeah, hello.
Yeah, sir. You have told about a lot about artificial intelligence and bioinformatics.
Yeah.
So can you share some details about the proteomics? Because most of the large molecules or small molecules, the research, whatever going on is mostly on the proteins.
Mm-hmm.
So can you able to share some insights regarding the proteomics, sir?
Okay, yeah, if you say, I assume that you want to know about the structural details, structural data analysis, how we are doing the structural analysis. Yes, in case of the drug discovery, when we go with the drug discovery, mainly we will see the protein structural informations. For example, the active sites or the cavities. There are different possible understanding of the cavity. The cavity can present in as like a groove or a cavity or a tunnel shapes, and that will help us to understand what type of drugs we are going to design. Whether the drug has a lot of conformational freedom or less, or we need to find a linear molecule, or we want to find it out a complex molecules. This all depends on the structural information of the protein. So we cannot just to see only the residual information or only the protein information, but also we need to look into the structural information, which gives an insight what type of small molecule structure is. because in simple terms, like you can see like the Emil Fisher lock and key model, like that we can see that how well the molecule can fit into the drug. It needs to adapt, or else the drug will look into the other protein. That's how we believe. There are several tools are available for this to understand. Molecular dynamics is also one of the interesting area for to address this, because protein is more. It is not a solid, right? It is you can make it like a gel, so it's more flexible. When the drug comes and binds, there will be a lot of induced changes also happen. So we need to consider that also. So NMR or the MD techniques will help us to address those informations. So with this you can get a lot of information about the protein side when you want to design a drug.
Yes, sir. Sir, you have shown some chart at the end of the session, like where are the artificial intelligence working. So most of them is focused on oncology and infectious diseases. Most percentage of work is going on oncology and infectious diseases.
Yeah.
But if you see the data, for last 20 years, we have very few antibiotics which come into the market. Previous 20 years, if you see, around 60 to 70 molecule came into the market, but last 20 years came to the market.
Yeah.
So even and as you told, the discovery period has been reduced 5 years to 1 year. You have exposed some data for that also. So when that technology came into place, it reduces the curiosity or intensity of the scientist. They are getting the data only based on the availability, and they are not doing any research like that. Or what is the reason behind that, sir?
No,
You
no,
can share
no.
your opinion.
Yeah, it's not like that. As I said, like it is vice versa. We will get the information from the hospitals as well as from the research, right? So we will know, we need to know how the drug is active or inactive in order to make sure that we need to optimize it. When the drug develop resistance, say for example, as you said, infectious disease or the antibiotics, if it develop resistance, then we need to see that what strain it diseases. Then we need to look into the mutation, what it happens. In order to answer your question, I will put this in other way. Now there is one more interesting field is growing. That's called where we'll address the possible predict the possible mutations. One thing in the protein level, where what are the possible mutations? Say for example, now this what we have, the COVID-19 is not the new one, right? Which is already was present before, but we don't know what in future what's again mutation will happen, what next mutation will happen. So if you can able to predict the mutations accurately, then also we can define predict the drug in advance. There is another field where why we have to study the protein or possible mutations in future. Instead, we can make the drug as a smart drug which can modify in its own to get into adapt itself to mutations. This is also another way we can. The researchers are going on where we can make a drugs which can change this, like how the proteins is changing when how it develop resistance. When the protein can able to develop a resistance, why not the drug can able to change its shape or characteristics in order to fit into the changed proteins characteristics, right? So that's how it works. So the data is important. Wherever you will the data we will get, accordingly we can get information. So did I answer your question?
Yes, sir. So I have another question. So whenever a technology intervenes, there is going to be a chance of reducing the human intervention in the particular field.
Yes.
So when it was coming to drug discovery, so when we are adapting completely technology oriented, like artificial intelligence or bioinformatics, there is a chance of reduction in the employability for the scientists. And what is your opinion on this?
Yeah, true. True, what you said is right, but we cannot do anything because now we are already they are testing the driverless car, so it means that a lot of drivers will go for a jobless. But they have to we have to adopt into some other ways how we can train. Yes, that job will go off, and they have to find it out the other way. And anyway, human intervention is needed, but as you said, the numbers will be reduced drastically. Yes, there will be a lot of job cuts, but there will be a more the people needed in the AI people who can develop the model. The model cannot develop its own. The people they need to shift the domain from the conventional discovery where it can be replaced by the machines or the models can be they have to shift to the place where they can make the models. There they have a great opportunities and much better options also. Existing people maybe, yeah, there will be a problem, but for the newcomers, it will be great because they will come as a trained people to develop these models, then it won't be a problem. Who already trained will be an issue, but anyway, it's not going to happen overnight. So the next generation, when it comes, then that field will be adopted. So I think there won't be, initially there will be a chaos, but later it will be balanced. This is how all the technology development happened so far. So same to AI also.
Thank you, sir. Because the question I have asked based on my interaction with our Pharmacy Council President, Dr. B. Suresh, sir. Sir, he came here to inaugurate a webinar 3 weeks back. During that time, he shared input regarding changing environment in the pharmaceutical industries. The top 20 manufacturer of India. They formed a forum and they have planned to introduce artificial intelligence and some automation in the manufacturing process to reduce the quality issues as well as to enhance the manufacturing processes. So after within 5 years they plan to implement those things. So being a bioinformatics and working on artificial intelligence, if our students want to counter these things, what are the skills they have to develop or what are the areas they have to focus on? Based on experience, can you share the things?
Yeah, so they can mostly for the artificial intelligence, again, the programming, Python programming is important, and also they need to know more about statistics and mathematics in general. Statistics is important, and if they concentrate on biostatistics and statistics and some programming skills, develop some programming skills, it will be greatly useful for them to be a trained pharmacist and AI pharmacist, which will be a great demand, because it's a very deadly combo. If a person, most of the AI people or the machine learning people who works in machine learnings are mathematicians. They are very good in developing models, but they don't have any idea about pharmacy. If a pharmacist or a pharmacy student who is working in AI, they know they know the AI, they know the maths, then they know the pharmaceutical background they have. Then it is a great successful combination. They have a great demand, so they have to concentrate on mathematics, statistics, and programming.
Thank you, sir. Any other questions? Any other questions? Sir, I think we are at the concluding part of the session, sir.
Okay.
So thanks for sharing your valuable experience with our students, sir. Like it is a new area, bioinformatics and artificial intelligence. So up to now we have thought it was more towards engineering students. So I think your session will be definitely an eye opener and it will provide a lot of information regarding their future career selection, sir, because it's one of the developing area. So I think some in future interaction with you may help our students. If possible, can we invite you for our future session also?
Yeah, sure.
Little more intense session on this artificial intelligence, sir, because it will helpful for our students, sir. So, sir, thank you for kindly accepted our invitation, sir, and shared your views on artificial intelligence and bioinformatics, sir. Thank you, sir.
Thank you very much. Thank you very much.
And so, if you have any plan to visit your native, sir, whenever you are visiting India, if you let us know, we
Sure.
can invite you to our invitation, sir,
Okay.
institution.
Thank you very much for the invitation. We'll come after this
Because
coronation.
yes, sir. Because the direct interaction will have more impact than this interaction through web, sir.
Yeah.
Yes, sir.
Okay.
Yes, sir. So thank you, sir. Thank you for spending your valuable time with us, sir. And your final thoughts on skill development part of our students, sir. What are the things they have to focus? Because you have been academics for 10 to 15, 11 years,
Yeah.
and you have been workers postdoctoral fellow in different countries. Now you are in an industry, so you may have some opinion regarding the academic and industry collaboration, sir. So what are the things students should do? Those things can you able to share? It may be useful for our students, sir. Thank you, sir.
Yeah. Okay. Yeah, since I'm also basically I'm also a pharmacy student, so I can understand what we are. What I can say is like the time when we studied pharmacy, we don't have that much technologies developed. Like now we have Udemy, Coursera, like many online platforms where you can easily learn the advanced fields. We no need to rely only on the textbook, right? So we have a lot of resources and a lot of courseworks which you can directly, for example, tomorrow if you want to study with the Harvard or Cambridge, you can just join the course available there, Udemy or Coursera. Some of the courses are free, some of the courses are very charging very less. You can choose your choice. Of course, you no need to be in one. Yeah, you can finish your courses, pharmacology, pharmaceutics. It's all important. Apart from that, if you are interested in programming or in machine learning or statistics or anything else, you can develop, because the future is not going to be only on the only for those who only knows about only the textbook and the traditional knowledge. We need to have we need to adapt ourselves to this, because every pharma industry is adopting AI. Every pharma industry is going with the machine learning approaches. They don't want simply to waste time and money just because you are very good in pharmacology and you have scored S grade in pharmacology and you have studied you are very good in pharmaceutics, everything. So apart from your textbook knowledge, and also we need to understand what is actually happening in the real-time scenario. You can also take some courses. Online courses are available. You can register and then you can take a course, and you can also do a lot of internships. The advantage of, say for example, take my company Pharmcadd. You can just Google it, Pharmcadd, and you can see my name and profile also there. You can contact me for anything if you want to know academically. I can also help you. And one more thing is like many of the companies like Pharmcadd like companies are the companies which works on the AI based or online based bioinformatics related works. You can do internships online. You no need to go there and do, right? You can collaborate with this company or with the scientist, and you can do the a lot of projects, mini projects and internships, everything online. Sitting in your home, do with a simple computer or laptop, you can work with them. You will get new ideas and practical experiences which they gained over a years. We can get it in few months. So that will be useful, since now most of you are in the lockdown, I think so, in India. So now so you can use this time to parallelly join multiple courses or communicate with several scientists available present or in the industry people. Like you can you can join with them to help them, or you can learn from them to get trained in the real-time environment. So that will be more benefited for a pharmacy student in the future. Thank you.
Thank you, sir. Thank you very much. Hope to meet you soon in our institution. Sir.
Yeah, sure. Me too.
Thank you, sir. Take care,
See
sir.
you.
Bye, sir. Bye.
Thank you. Have a good day. Bye.
Okay, bye, sir.